Good morning, everybody. My name is Guy Levy, and today I'll present a technique that we developed for studying the locomotion of the tobacco hornworm caterpillar, Menduca sexta. The way animals control their body is simply amazing, and this is why studying motion control, which is what we want to study to understand how they control their body, is very interesting by itself. And also it has an added value because sometimes it can be used as a source of inspiration for the development of control mechanism for artificial systems. And what is studying motion control? What do we want to discover? We want to better understand the strategies of the nervous system that it uses to calculate the correct sequences of commands to the muscles to generate efficient motion. We want to understand the motor programs, the algorithms that are inside the nervous system. We want to know what the animal can sense and how it uses the sensory information to control its body. We cannot directly access the nervous system to read these algorithms, so we need indirect ways. For example, one way is by observing and analyzing the movements of the animal. For example, here, the walking of this dog teaches us that it can sense the friction coefficient of the substrate, and when it realizes that the ground is slippery, it decides to go in this amusing way that we see here. How do we get the data? How do we obtain the data for motion analysis? One way is by tracking points on the body of the animal as it moves. And how do we track the points? A commonly used way is by video recording the animal, then extracting separate video images, and then labeling the points of interest on the video images. But this manual tracking limits our ability of correcting things during the experiment, because some things that are needed to be corrected can be noticed only after the analysis. And also it is very slow, even when a high-speed camera is not required for our experiments. Let's just make a quick calculation to get a feeling of how slow it is of manual tracking. So let's say we want to track 6 points on the animal's body, and so it means that each video labeling of each video image would take about 12 seconds. Let's say 2 seconds per point. And let's say we are using a standard camera with frame rate of 25 frames per second. It means that we'll have 300 seconds, which are 5 minutes to label 1 second of experiment. It means that in order to label 1 hour of experiment, we'll need 5 multiplied by 3,600 minutes to label. And if we want to do it in hours, it would take us 300 hours only to label the 6 points that we want to track without yet doing the analysis. So both Professor Trimmer, my advisor, and myself did manual tracking in the past. He did it for studying the crawling in the Menduca, in the caterpillar, and I did it for studying coordination in the octopus. So we knew how unpleasant it can be and how slow it can be. So we decided to create a new method, an automatic data acquisition system, preferably in 3 dimensions, and that would do all of the analysis in real time. I must tell you, at the time it seemed kind of like science fiction, but we wanted to try it anyway. So we wanted to base our setup on the Vicon. The Vicon is a commercial device designed to capture 3-dimensional human motion in order to embed special effects in films and video games. The participants are surrounded by cameras that are sensitive to infrared light. The strobes on the cameras emit infrared light that is reflected back by the retroflective markers that are attached to the dark clothes that the participants are wearing. The system is using the reflected light to follow the movements of the participants and reconstructs them in the computer. The reconstructed motion is used to create the special effects. We naively thought that we could mimic this setup by using miniature beads that would serve as infrared reflective markers. But soon enough, we understood that the beads were too small and cannot be reliably identified by the Vicon. Then we thought, why do we need to use markers that are reflecting the infrared light? Why not use markers that are emitting infrared light? We tried to look for a light-emitting diode that would be small enough to serve as a marker, and we found that Osram company manufactures very tiny LEDs This is how they look. This is how it looks under the microscope, next to the bead that we initially intended to use to glue onto the cuticle of the caterpillar. I admit that the LED is larger, but we thought it would work. We succeeded in soldering these into a suit with a very fine wire, kind of a suit, and stitched it to the cuticle of the animal, of the caterpillar. This is one stitch. You see the stitch on the right and the forceps on the left. It was very, very shallow under the skin, and it's done under anesthesia. And it seemed to work, because this is how the caterpillar looks with our LED suit. Maybe we can turn off the light above the... And it seemed to work, but only for a moment, because the animal was not really behaving normally. The LEDs were too large. And then we thought, why do we have to use the LED as it is, as it's sold inside the package? Why not use the LED itself from within the package? Because this is not the LED. This is the LED inside the package. This is the LED. It's much smaller and thinner. And then it's not intended to be extracted out of the package. They sell it as a unit. But we tried anyway and succeeded in extracting it. And if this is the LED with the package, this is how it looks under the microscope. We succeeded detaching it and isolating it, and it was much smaller. And we succeeded in manually soldering them into a suit and stitch them to the caterpillar. This is how one such extracted LED looks when it's stitched to the cuticle of the caterpillar. This is under the microscope. And this is how an anesthetized animal looks when it's wearing our LED suit. And this looked much better, because if this is the first LED suit of the LEDs, of the original LEDs inside the package, and you see how it's said when it's crawling with it, this is how it used to crawl when we used the new LED suit with the extracted LEDs. So it was much better, but it could still be improved, because if this is the LED that we used to extract with a square of 0.8 millimeters, we found something even smaller of the same company with the same wavelength, which has a square of 0.3 millimeters. And we succeeded in extracting also this and soldering it into an LED suit. Next, we understood we needed a treadmill to keep the caterpillar inside the scene. This is always true, but with real-time analysis, it's critical. So this is the evolution of our treadmill. Initially, we used the cup as a treadmill. Then we moved up to a petri dish. Then we added the option of wiring it to enable turning on the LEDs. And then we thought, why rotate the treadmill manually? Why not use a motor to rotate it? And why not use a controller that's hooked up to the computer to rotate it according to feedback that it gets about the position of the caterpillar? And then it could be held in a fixed position, in the fixed orientation. You see how it's rotating? And then we did some more improvements, like adding this vent that motivated the caterpillar to move. And we added this gear to make the rotations more soft, more gentle, so they won't influence the behavior of the animal. And here you can see the experimenter can control the slope in which the animal is crawling. This is 180. Okay, so by then we could place the caterpillar dressed with our suit in front of the vital cameras, which could detect the movements and send them to a computer that by now could present the movements. Then we realized that although the only information needed for the analysis is the position of the markers, we cannot do practical experiments without adding a video, because it is needed for the context. So we added a webcam to our setup. And by using some linear algebra, we succeeded in projecting the positions of the markers onto the video. Then we could create this reconstruction panel, which shows the position of the markers relative to the treadmill. And we could add this analysis panel through which we control and manage the experiment. Sorry. So we had this very nice experimental setup, but it was working very slowly. it could handle about one sample per second, and then we needed to improve the performance. So we changed our software to include parallel processing with a combination of MATLAB, which is very user-friendly but not very fast, and C++, which is very fast but not very user-friendly. And we separated the burden across computers, and this dramatically improved the performance of our setup, and now it can handle about 30 samples per second. Now let's go through the complete setup and see what it can do. These are the Vulkan cameras with additional webcam, which are altogether video recording the animal as it crawls inside the treadmill. This is our graphical user interface, through which we are controlling the experiment. And let's, for just 1 second, see it alongside with the original video of the caterpillar inside the treadmill. The experiment can be controlled, for example, by setting in which orientation the caterpillar will be crawling. For example, here I'm about to change the crawling orientation from a slope of 0 to a slope of minus 45 degrees, and the treadmill automatically shifts to holding the caterpillar in this orientation. Using the analysis panel, I can choose which steps that were automatically detected by our system to accept and which to reject. Every experiment is saved into a hard drive, so it can be repeated. So we can repeat the experiment in offline mode, do more analysis, or see it again, or for other reasons. In offline mode, I can decide which part of the experiment to repeat in any pace I want, go back and forth. Now, do you remember we said it would take about 300 hours to label 1 hour of experiment without the analysis if we are using manual labeling? Then here you can see I did the automatic process, this is the total time. It did the same thing in real time, in 1 hour, and it collected more than 200 steps. And here I'm showing only a small example of what we achieved of this one session of experiment. This is the result that we didn't really intend to do, but I can show here that we are measuring the horizontal velocity of the pro legs, the black line, and the vertical velocity, the gray lines, in different segments of the body. And here we can see a phase difference between the segment A6 and A3. And so we have this marvelous setup with which we can do very precise 3-dimensional real-time tracking and analysis of the crawling. And it can be simply adapted to be used in many other variations, including other animals. I can confidently say that this new method we developed would significantly elevate the study of motor control to a higher level. And here I want to thank my advisor, Professor Barry Trimmer, for generously granting me access to his amazingly extensive knowledge and his extraordinary insights. And I would also like to thank this group of young and very talented people with whom I'm privileged to work. I'd like to thank the National Science Foundation for funding this project. And I would like to thank you, ladies and gentlemen, for listening to my talk.